From Microbe TV, this is Immune, episode number 10, recorded on July 17th, 2018. I'm Vincent Racaniello, and you are listening to the podcast about the body's defenders against disease. Joining me today at the University of Maryland, Cindy Leifer.
Hi, welcome back.
We are in person.
We are all in person, and I have met Vincent before, but I am just meeting Steph at the meeting for the first time in person, and it's been lovely. And we're at College Park, where
Maryland
is my alma mater. I went to school here, so it's
been
fun. Yes.
How cool! This campus.
It's
like yesterday, right?
No, it has changed so much that I barely recognize some of it. So, but it's been a lot of fun, a lot of nostalgia.
Cool.
Walking around, yeah.
Also joining us from Worcester, Ohio, Steph Langel.
Hi, everyone. Again, similar to what Cindy said, it's great to be in person.
First time, right?
This is my first ASV. I've mostly in the past gone to immunology-focused meetings.
Me too.
Right?
Me too.
Obviously, we are an immune, so we're more immune-focused. But what I've loved about this conference, firstly, the topics. I mean, there is viral mechanistic workshops, but there's a lot. I mean, I went to an adaptive immune workshop 2 nights ago, so there is a lot of immunology throughout. And Cindy and I were talking, and we just feel that this meeting, there is a collegiality and a warmness amongst the people who are here. As a grad student, I definitely feel that from different places I've been, so I appreciate it a lot.
And this is our first immune where we are together in the same room.
That is correct.
And it's number 10.
It
which
is.
is pretty cool.
Milestone.
So you know what they
Yay.
say in podcasting? Most fail at number 12.
Uh-oh.
So we're getting there. We're not going to fail.
Drum roll.
Oh, we're not going to fail.
We're not failing.
We're on the upswing.
We're getting there. And
yeah.
and plus, and on top of all that, we have our first guest.
Yay!
She is from George Washington University School of Medicine. Rebecca Lynch, welcome.
Thank you so much for having me. I'm excited to be your first interviewee.
And you're right here in the area. You're in Washington, D.C.
Yes, I took the metro.
You know what they say, when you go to a meeting, you shouldn't commute from home because you're not really at the meeting, right?
Very true.
But it's cheaper.
Right,
to take
exactly.
That. So you take the metro to where?
I take the metro to the College Park, University of Maryland metro stop, which I had actually never done before. I'd never been to this campus before. And
where is that stop? Where is it located?
It's about 5 minutes drive down Campus Drive here, so it's very close. And I've been Ubering in my ignorance from the metro to here, and today I paid attention and realized I could be walking.
There's a shuttle. Yeah.
There's a shuttle, and I could be walking.
Right.
It's hot, though.
It is hot.
Very warm.
And it's hot in this room, unfortunately, but it's quiet, which is what we need for podcasting, right? So we're all going to be very hot by the end of this, but that's okay.
There was blowing a lot of hot air. Right?
There is another. There's a room behind us that we could have used, but there's too much air noise, and I wanted to have a nice, quiet, pristine podcast. So number 10, our first in person, our first guest. That's a great trio of things to do, right? So ASV I've been to many times, and I love it. I think it's a great meeting. It's collegial. A lot of young people here,
Absolutely.
right? This is a student postdoc focused meeting.
It is.
Right. Yeah. There are some PIs who come every year. They're great. They're really great but most don't. You know, it's the usual thing. They don't come unless they're paid for, unfortunately, unless they're invited. And that's too bad because I'll come to this forever because I love it. And I love walking around and talking to people. And I'm kind of in a special situation because people know our podcast and they come up and thank me.
Oh, you're a celebrity.
You have had a lot of
Every
selfie
time
requests.
I turn around, selfie.
And I
did have one selfie request, so I was like,
yeah, it's great. I love it. I think it's really cool that science comes around a podcast, or in my case, a lot of people have told me how they use my lectures. I put my lectures online, how they use them. I just think it's great because you don't know. You put stuff out there and you kind of don't know who's listening. Right. And so my strategy is I just stand in the middle of a room and look friendly and people will come up. And that never happened before in my life before podcasting. No one would, maybe because I was scowling all the time, right? But I love it. I just love this meeting. I think it's great. And
I have to say that, I was impressed, surprised, and having a great time with how much immunology is here.
Yeah, it's a lot. Yeah.
Has it always been that way?
Always,
or is
yes.
it? Yeah.
I'm glad you. Yeah, I thought you would be okay.
I love it. I actually like this better than some of those immunology meetings that I go to. It's
great.
just fascinating. Great science, really.
So let's talk a little bit with our guest and. Starting off, where are you from? Tell us a little bit of your backstory, your back history.
Okay. So I grew up in a small town outside of Boston, so I'm originally from Massachusetts.
You have a little bit of an accent left, right?
Oh,
very little. Very little.
Hide it. So most people can't hear it. So I actually got started in science looking for a summer job. So I was going to school in Boston for high school, and sort of a friend of a friend. I took a biology class freshman year. I enjoyed biology, thought science was cool, and it was just looking around for a summer job. Randomly, through a friend of a friend, heard that someone would be willing to take a high school student into their lab, and I started learning how to do ELISAs for the summer for free.
Oh, great.
Yeah. So
when was this, in high school?
This was in high school, so I was actually trying to remember what year that would have been, 1995, 96. So I learned how to do ELISAs, and I've always tried to remember that chance to just have that high school internship changed my life. It's just funny how life works, and I still do ELISAs.
So you loved it?
I loved it. I loved following protocols. I loved the colors that turn at the end of the ELISA, getting the results, learning how to graph them, what they meant. And so then that turned into a job in Summers in the Future. And so it just really shaped my life.
Was that an immunology lab? Was it a virology lab?
It was an HIV lab.
Okay.
So I really I've been focused on antibodies and HIV since high school. What I enjoyed was that I felt, even though I was doing this bench work, that there was some bigger meaning behind it, and that HIV was an important problem. And the more I learned about actually immunology and virology, because I started not knowing anything, I just was pipetting and following a protocol, how interesting HIV is as a virus.
And did you work in labs all through college as well?
So a couple summers came back and worked in the same lab. It was really a touchstone for my life. I worked in a couple labs in college, but they weren't HIV antibody labs, and so I did not enjoy them so much. And then after college, I worked through the post-bac program at the NIH for a year.
When were you there?
So it would have been 2003, 2004.
I was there.
Oh, really?
Yeah, I was. Yeah.
I
left in 2005. So I was there between 2000 and 2005 as a postdoc.
Oh, wow.
Yeah, I went to all the immunology retreats and things.
Oh,
yeah,
cool. I was working in Ira Berkhauer's lab in the old FDA building, sort of in the middle of campus.
Cedar or Cedar.
Yes.
Yeah. My first postdoc was in that building.
Oh, no.
And then I moved over to Building 10 to NCI.
So maybe you even crossed paths.
We may have.
From building to building or something.
Completely.
Yeah, probably.
That's funny. Actually, so it was very funny when I came back from my postdoc, I can't even do the math, 5 or 6 years later, to walk that same path. The NIH, it changes sort of, but not really.
Right, yeah.
Walking the same path to work. But I did.
It's an amazing immunology community there.
Yes.
It's just so many really smart people doing great stuff.
Yes, I miss it. And I almost think that when I was there, I didn't appreciate, you sort of appreciate it, but when you leave, you really appreciate what an amazing community is there.
Where did you go to college?
Oh, I went to Yale University in Connecticut. So that's where I took my first immunology class.
Who taught it? Do you remember?
Yes, Kim Blankenship. And I didn't realize at the time that the Janeway textbook we were using,
was
So
Janeway.
it took me so long to make that connection of the amazing immunologist at Yale. I was a little bit oblivious when I was an undergraduate.
Yep.
So that was wonderful. Yeah, I remember I don't know if every scientist has this, but the debate between being a medical doctor versus a PhD, because you think being a medical doctor is the more practical pathway in life, but I just didn't really like people that much and really liked bench science. So, in the beginning, you have to fulfill all your biology class requirements. It's full of pre-meds. I just didn't enjoy those big intro classes. But then by the end, junior, senior year, you're taking the small classes in immunology. Yeah, it's overwhelming, but I just love the topic so much.
It's a great immunology faculty there at Yale, right?
Yes.
It's awesome. It's awesome. I mean, our chairman came from there, Shankar Ghosh.
Mm-hmm.
You have Akiko Iwasaki.
Yes.
Ruslan. Yeah, you can pronounce that last name.
People often mix it up with Mechnikov. Who was,
but you probably didn't encounter any of those individuals, right?
I'm almost positive some of them were lectures in my immunology class, but again, I was totally
you don't even
oblivious.
know that you're being taught by such famous
and
people.
then in retrospect,
so you're like,
back, oh my gosh, yeah.
Well, I remember I had Jerry Fink in college for a genetics course.
Oh wow!
I had no idea, and then many years later, he's director of the Whitehead. I said, "Wow, that's pretty cool." So when in college did you decide to get a PhD?
I want to say about halfway through sophomore year.
Well, that's early.
I think I knew the whole time, but it was sort of gathering the courage to tell my parents that I am not a pre-med. I just can't.
You did better than I did.
Did it take you longer to?
Well, my father was a doc, and he wanted me to. And I went through college. I could never say no, so I ended up graduating with nothing to do. I hadn't gotten into medical school. I only applied to one place, and I couldn't say no.
Right.
Well, that was how you said no, right? You applied.
Like my wife says, I'm passive aggressive. Yes. Instead of saying no, I just do some other things. Yeah.
But so did you know yourself that you didn't want to be an MD?
Yeah. I didn't want to treat patients.
Right.
I mean, I saw what he was a surgeon. It was horrible hours, and he used to tell me what he did. I just didn't want to do that.
Right.
And so, but I couldn't say no. And I didn't know what else to do. But I was majoring in biology, so as you may have heard at the keynote, eventually it worked out, right?
Right. A little bit. Yeah.
Where did you go to graduate school?
So I went to graduate school at Emory University. So I took 2 years, and I was a lab technician. And actually.
Oh, I thought you were going to say you got your PhD in 2 years. I'm like,
okay, wow. Oh gosh, no.
You took 2 years to decide what you were doing. Okay,
right.
You worked,
yes.
You worked at Emory as a tech?
No, I also had this amazing. I have fallen into these amazing opportunities. I decided. So I did one summer in Ira Berkauer's lab as a post-bac at the NIH, and then I decided that I wanted to live in Manhattan or New York. I ended up living in Brooklyn because I couldn't afford Manhattan. And so I just applied at the Rockefeller University to be a lab techniciin.
And who did you work with there?
In Ralph Steinman's lab.
Oh my gosh, how lucky for
I know.
you.
Again,
such a wonderful individual.
At the time. How amazing.
So when were you there? Can I ask it? Because this is funny.
2004 to 2005, like that. It was just a short time where I was interviewing for graduate school, and
that's where I did my PhD. I was in New York City, right in that area. And I'll tell you, so you've been now in two of the, I think, amazing places to do immunology. One is in New York City in that area there with Sloan Kettering, Cornell, Rockefeller, and the other one is at NIH. So I've been in both of those places. Both of them are amazing opportunities.
Yes. Yeah, beautiful. And just the colleagues, just being able to walk around and talk to different people, and everyone is so smart and interested in their topic. Those are both two special campuses. But again, those two things I fell into when was young and didn't appreciate at the time probably how cool working there was. So then I started in graduate school at Emory.
So Emory's not a bad place for immunology either.
No, that's true. That's some amazing immunologists there. Yeah. And what did you study there? You're going to say HIV, antibodies?
That's the answer to every question you're going to ask me about my career. So I worked in Cindy Jardine's lab. She was a postdoc with Eric Hunter. They had both, so she was starting her lab. So she was a brand new assistant professor who was starting her lab at Emory, and they had moved from UAB and had started really interesting to me studies on transmission, where they were using samples from a cohort in Africa where heterosexual mismatched partners, so one partner has HIV and the other one doesn't, discordant couples they were called, were followed and regularly sampled in order to intervene and try and counsel them to not transmit HIV to their partner. However, because of this intervention and sampling, it allowed us the opportunity to look quite frequently at samples from people who are newly infected with HIV. Mm-hmm. So with Cindy, I started researching in her lab what the natural antibody response early in infection is to HIV infection in this African cohort. And Cindy taught me a lot about just basic virology. You know, we were doing molecular cloning and mapping epitopes and just basically swapping areas of the HIV envelope and seeing, you know, do serum responses still respond? Do they not? So that was really cool. And then that led me to, so we had these graduate school seminars where the students voted to have people come down, and we had these guest speakers. And Dr. John Mascola came one time and was talking about antibodies directed specifically at the receptor site of HIV. And I thought his work was so cool because it was even more translational and vaccine focused than what I was doing in my lab. And so an opportunity to postdoc in his lab came around, and I was so excited to do my postdoc there. And then that was at the NIH. And that, again, was an amazing opportunity.
And so after that, you came right to George Washington, is that right?
Yes. So while I was there, I decided that I wanted to pursue. So, I have to be honest that I debated when you're doing your postdoc. You know, when I was in graduate school, I was just really focused on getting my postdoc. And I got this postdoc of my dreams. I was so excited doing the work. And then you're sort of like, what am I going to do with the rest of my life? And at that point, I'd always thought I wanted to be a PI and go into academics, but I really did consider different options. The NIH has a lot of options with being a staff scientist, and sort of you don't have to write grant, you know, so sort of where do you envision your career going? Where do you want to focus? What kind of science do you want to do? What are the things you are willing to deal with to be a PI? And I decided that I wanted to go for it. I wrote a K-22 that was eventually successful, and that really helped me transition and get a job. But also just when I started talking to people, how do you get a job as a PI, as an assistant professor? And basically the answers were you apply to tons of positions. It's a numbers game. But also that everyone had this unique story where I talked to this person, there happened to be this opening, and these stories didn't seem widely applicable. It seemed that worked for you in this one specific case. And then what happened to me was sort of the same thing where George Washington University is looking. They were starting an HIV CFAR, which is a Center for AIDS Research. And they were really interested in putting money in this and building an HIV-focused community in Washington, D.C. Washington, D.C. is a big site of the epidemic, and there wasn't a lot of collaboration within the HIV research community in Washington, D.C. And to me, that was an exciting opportunity. But again, that just happened to be that they had a new department head in the microbiology and immunology department who was looking for HIV researchers. At the time that I was looking for a job, I didn't have to move.
Nice. How long ago was this that you came?
So I started at George Washington as an assistant professor 2.5 years ago. Which means I sort of probably started applying for jobs 3 to 4 years ago.
Did you already have the K-22 when you went on the job market?
Yes. So the K-22, and I have to be honest, I think the mechanisms have changed through the years, so I'm not sure what it is now, but at the time that I applied, you had to apply within 4 years of graduating from graduate school. And then and they split it into 2 parts, and once your first scientific application was accepted, you then had 1 year to find a job, and then
same thing. I got a K-22, and I did
the
same thing.
Yeah. And that was a big help. I think if I didn't have that, it would have been much harder. Yeah.
Tell Steph, now you know what you have to do.
Well, I was in this discussion, the one discussion table, they have a bunch of them for trainees, and we can go talk about a bunch of different things. But Dr. Vera Tarakanova from Milwaukee Medical College talked to us about the postdoc to PI transition. And she was excellent and very specific in telling us, there are things that you need to get if this is your goal. And the K22 is one. The K99 is, of course, another one. I know that that's a 1% pay line, so it's a lot harder. But can you talk about the timing of all of that? I mean, you said that you weren't sure if you're going to pursue it. So when did you make the decision, okay, I'm going for it, then the K22 got written? Is that the steps?
Not necessarily. I decided that I wanted to keep my options open, and timing is so important, and that's what I became aware of, and that's something that, you know, I encourage everyone to just talk to people, because that's how I became aware of the timing of the K22 and worked out that even if I didn't want to be a PI, to keep that option open, I needed to have this, you know, these things in place. And because there's a time limit from when you graduate to graduate school, you really need to be thinking about it before you're ready. And so a big piece of advice I would say is I have had people, and one friend in particular who's a couple years ahead of me in my career, and she provides invaluable advice because, you know, it was pretty recently that she was in my shoes and she got a K22. I also have to say that the NIH has an amazing office for training and education. And so if you reach out to them, you know, they have such a great perspective because they've seen so many people apply for the K Awards. And so that was also an amazing resource. And I'm sure there are career development offices like that in lots of different places. And so basically I met one-on-one with them. I went to seminars about K22s, you know, learning your options. Because I know that at the NIH I was probably more limited for what I could apply for. So that's one thing about doing your postdoc at the NIH versus everywhere else, is you might have more opportunities for applications for funding. Right. So I was limited to the K22 and the K99. And as you said, there are different pros and cons. So again, talking through, you know, mentored versus not mentored, the pay line, etc. I decided the K22 was a better fit. It was less money, but a higher pay line. And then what I also had to factor in was not only the time from when I graduated graduate school, but also that you can resubmit it twice. So allowing for not getting the grant the first time, getting reviews, responding to reviews. And so when I worked it back, I think I started applying or thinking about applying, you know, 3 years into my postdoc. I was loving my postdoc. I didn't want to leave, you know, so in my head, I still didn't know what I wanted to do. I just wanted to do my postdoc, but I wanted to set myself up so that at 5 years, I was in a position, you know, to have opportunities. So I would say,
was that a similar experience for you, Cindy?
Yes.
Yeah.
Yes. Yeah. Right. Exactly the same. It was a grant writing workshop to prepare the K22 and submit it, and yeah, exactly the same.
And there are some people in our table discussion, and who may be listening to our podcast, there are USDA. So if you're more focused, like plant viruses, they don't always have that. So there's also NSF and USDA postdoctoral, that if you're applying maybe for a different aspect of science, there's those options as well. But
now, did you write yours on these broadly neutralizing HIV antibodies? Were you already starting to think about that? And can you talk about that a little bit?
Yes. So I wanted to capitalize, and I think this is maybe an issue with everyone when you're doing that transition from postdoc to PI. You want to capitalize on your experience, but also create your niche that's different from what you were doing in your PI's lab. I was really interested. I mean, right. So I've already admitted that my entire career has been antibodies against HIV. So I was very interested in broadly neutralizing antibodies, and that was the work I'd done in John's lab, and especially antibodies that target the receptor binding site of the virus. And so I wrote my K22 on a project that was partly a project that I was finishing at the end of my postdoc, which was a clinical trial using broadly neutralizing antibodies in people who were chronically infected with HIV, and how does the virus escape when you stick in a heterologous antibody that this person hasn't co-evolved with, and then building into an in vitro system, actually still working on, but basically can you try and look at escape from antibodies in vitro systems so that you don't have to always do these clinical trials with people? How do you recreate virus diversity that's replication competent in an in vitro system? I've always been interested too right now in which combinations of antibodies are better. Do you want to target one site with a bunch of different antibodies, which no one seems to think is a good idea, but I still am interested in the concept, versus targeting a bunch of different sites on the virus at the same time?
So one of the things that I don't know if all our listeners know is exactly what is the broadly neutralizing antibody? Why do we need those? So we're immunologists generally, and we know the importance of antibodies, but why these specific ones?
That's a really good question. And I think even with people in the field, when we work on broadly neutralizing antibodies, we need to sort of think about it every now and then, what we're really talking about when we say these phrases. So HIV, one of the reasons that I'm so interested in it as a virus is its incredible genetic diversity. So when a person's infected with HIV, now we know oftentimes they're infected with one genetic strain of HIV. And these days there are many treatment options, but just in general, if you don't treat the HIV, your immune system cannot clear the virus, and the virus rapidly grows inside you and escapes. So you develop T-cell responses against the virus, and you develop antibody responses against the virus, and the virus can rapidly mutate and escape. And so what happens over time is you have lots of genetic diversity in each person infected with HIV, and that sort of generally at a population level ends up meaning that there are a lot of different genetic subtypes and species around the world. And so then when you think about treatments or vaccines that need to be used at a population level, how do you protect people from all these genetic diverse species of HIV that are circulating at the same time? And so then from there, you think you need when we're thinking about the antibody field, you need antibodies that can recognize generally conserved parts of the virus so that they can, so we were focused on neutralization. So antibodies that can recognize, bind, and block infection of different virions that express, proteins with different amino acids and different sequences. And so one way to do that is for the antibody to recognize a conserved site on the virus that doesn't change so much over time, or you need antibodies that can tolerate lots of different proteins. So maybe the site isn't super conserved, but the antibody has a lot of wiggle room in how it binds and it can tolerate. So those are sort of two main ways that antibodies can be broadly neutralizing, but basically it just means that they can neutralize, recognize, and bind lots of different genetic diverse HIV strains.
So if that's a really good thing for our immune system to do, why don't we do that normally? So we often, our body creates antibodies against parts of the molecule that do mutate pretty frequently, right? That's why we're not successful at controlling this particular virus.
Right. So I would say that each person is so different. So you have the antibody and the virus within that person that are co-evolving. And so you develop antibodies over time that are neutralizing in the sense that they neutralized all of the archived virus that you've seen, and the virus always escapes. And so it's this constant back and forth within the person. But that doesn't mean that that antibody can then recognize all the evolved viruses from other people. And that's really an in vitro artifact or something that we've come up with. That term or that ability is not something that helps you as a person. I don't know if this is making sense. Yeah. So it's sort of we're looking for these broadly neutralizing antibodies that sort of confer no protection or help usually to the person that they're from, but that if you could get a vaccine that would help people make this antibody before they're infected, or you could give people this antibody before they're infected, maybe that's a different scenario where...
How do you identify these broadly neutralizing antibodies?
So that's another good question. So in general, I would say that there are two main methods that people use, and one is first, I would say most people, you screen people. So very few people make these broadly neutralizing antibodies naturally. Again, it's sort of this category that we've come up with that's useful for us as scientists, but within a person, it doesn't really matter. So most people screen people's serum to see if they develop antibodies that have the ability to neutralize lots of different viruses. And there have been a couple different studies that show, in general, it's just this linear scale that 20% of people can neutralize 20% of viruses, and vice versa. I think I might have said that wrong. 50% of people can neutralize 50% of viruses in vitro. 10% can neutralize 90% of viruses. So it's this scale where you get on the end, and when you're looking at for super broad neutralization, so say 90% of all the viruses that you've tested in vitro, very few people can do that. So you focus on those 10% of people that make these really great antibodies. Then I would say in general in the B cell field, there are two ways to go about pulling out and identifying these antibodies. And one is to say we're going to do it unbiased. We know this person makes these great antibodies. We're going to sort all their memory B cells, let the B cells generate little bits of antibodies in an in vitro culture, and then test that in our in vitro system to see which well matches this neutralization profile we're looking for. Another way to do it is to map, these people make these broadly neutralizing antibodies. Let's try and figure out using their serum what is the specific epitope of the virus that they're focused against. And then can we work with protein experts and recreate that epitope, and so that when you sort out memory B cells, you're actually just sorting that small population of memory B cells that sees that particular epitope that confers broad neutralization. And my work has mostly been with the latter.
How many different kinds of broadly neutralizing epitopes are there? Are there a few? Are there dozens?
So that's a good question. So when we give presentations, we tend to narrow it down. There are these certain sites that are targeted, and we can list them. But more recently, as the structure of HIV has become more and more well-defined, I think there's a general consensus that there are very few spots on the virus that are not covered in glycans, and that basically any spot that's not covered in glycan, in theory, you could get antibodies that neutralize. And again, so broadly neutralizing can also be somewhat subjective. Is it 50%? If you neutralize 50% of viruses, that can be sort of considered broadly neutralizing. Some of the most potent or the broadly neutralizing antibodies that people have really focused on do tend to target a couple different sites. And so one is the top of the virus, the apex. And then another site is called the V3 glycan. So it's variable loop 3 plus a glycan epitope. It's a glycoprotein. Another site that I've really focused my research on is a classic virology site, is the receptor binding site of the virus, which is obviously conserved because the virus needs that site to not change that much so it can keep infecting T cells.
And these are all on the envelope glycoprotein only, right?
Yes, and so we've only focused on the envelope glycoprotein because that is the protein that's expressed on the outside of the virus that antibodies can see.
Did you see the glass HIV here?
No.
Did you?
I saw your tweet of it, but I have not seen it yet.
Have you seen it?
I've seen the pictures only.
All right, so there's a glass artist called Luke Jerram that we've talked about a lot on TWIB, and he makes glass models of viruses. And there's one here. It's in the art acquisition part here. You should
I need
actually
to see that.
Go down one floor and over to this side. Okay. And the reason I'm pointing this out, it's beautiful. It's a glass sculpture. He's in museums, and these things are enormously expensive, right? But it's got the membrane inside. You can see the conical nucleocapsid, and it has glycoproteins, right? And I was with someone last night who works on HIV, and she said there are too many glycoproteins on it.
So funny. I was thinking in my head, how did he know how many glycoproteins to put on the outside?
Right, so it's very sparse, right, the number of glycoproteins.
So apparently that's another immune evasion technique, yes.
It's amazing, because most viruses are covered with packed glycoproteins. And you should look at it. You have to go look, because it's your virus. Right?
There's one in the Corning Museum of Glass as well.
An HIV?
Wow.
Same guy?
I don't know.
Yeah, this guy's very famous. He's in museums, and you probably would have to pay $100,000 for this thing. Can't afford it, but... And so that's what we're. So why are they so rare in people? Do we understand that?
Oh, the broadly neutralizing antibodies. That's a really good question. So I would say that one answer is that breath, the way we define it, does not help. It's not evolutionary advantageous to the person who's infected. So one part of a study that I did in John's lab was just looking at people that made antibodies against the receptor binding site. And not all of these people were broadly neutralizing, but in general, we were just looking to see if there was a correlation between people who make these antibodies and any sort of clinical variable, such as lower virus load. et cetera, and there wasn't. And so really, because there are so few people and we've just studied them individually, it's hard to say, why these people do versus not. But one reason that seems to be is for whatever reason that these people in an epitope that can be broadly neutralizing, there's a lot of variation. And so in that way, it sort of stimulates antibodies that can either tolerate a lot of variation at that specific epitope. So one paper that I focused on in John's lab was studying the person. So one of these broadly neutralizing antibodies is called VRC01. It was the first antibody from John's lab at the VRC.
Vaccine Research Center, right?
Sorry, yes, the Vaccine Research Center.
I didn't realize that's why it was named that. Okay. Yeah.
Yes. So so they found this antibody is very exciting, and it's gone into clinical trials, et cetera. And part of my research at the postdoc was to look at the viruses in this person and investigate how did the virus escape. So in theory, this person developed this great antibody. Was there any clinical benefit to this person? And so anecdotally, this person did have a slightly lower virus load than normal. So maybe there was some, maybe the antibody was applying some pressure to keep the virus load low, but certainly the virus was still there. It was detectable. And his virus escaped, and we could study that over time. And so we could see that this person it wasn't a seroconverter, but at one point this person had virus that was very sensitive to VRC01-like antibodies. Their CD4 binding site sequence was somewhat unusual, like the receptor binding sequence was somewhat unusual. It was pretty open. There's a glycan sugar that usually covers part of this important epitope on the virus, and it wasn't there, which is very unusual. But maybe one of the reasons that this person developed really great receptor binding site antibodies, and then you can see the glycan develops over time, and then other sequence changes develop over time, so that person's virus becomes resistant to this great antibody. But when you take the antibody out of that person, in theory, it neutralizes a lot of different viruses. It could be great. But also, I am guessing that you almost sort of trained the antibody to learn to deal with this glycan. So you have the epitope exposed. It induces a B cell that can see this really conserved epitope that's good. It brings the glycan in, and instead of becoming totally resistant, the antibody lineage learns to target this conserved epitope and incorporate that glycan as part of its epitope. So it's not resistant. It can still neutralize the virus. And that glycan is very conserved. So the ability to see the receptor binding site and that glycan and tolerate it is one of the reasons that antibody is broadly neutralizing. But then for that person, I don't know that helps them at all.
Yeah.
Now, do you know, are broadly neutralizing antibodies produced in pediatric HIV? Are any of the antibodies that you found from children?
So no, I've not focused on pediatrics. So I was listening to your immune on the RSV infants. And so I was thinking about this, and I didn't go back and read the papers, but there's a lot of work from Julie Overbaugh's lab at the University of Washington who does focus on mother-to-child transmission. And she has recent papers where they did find infants who make broadly neutralizing antibodies, which is very interesting because it's always been supposed that one of the reasons people make broadly neutralizing antibodies is because they've been exposed to virus over a really long time. There's also a number of people who are super infected who make broadly neutralizing antibodies, which means they're infected with one strain of HIV and then they're infected with another strain of HIV. What the role of having two different strains of HIV, if that makes generates so much diversity that helps people make broadly neutralizing antibodies is another hypothesis.
But in that RSV paper, they said there's no germline HIV antibodies, whereas there are RSV. Right. So the fact that you get them from young children means they've been infected a lot, right?
But I'm curious then, are those broadly neutralizing antibodies that were passively transferred from their mothers in the placenta that happen to still be in circulation? Or are they endogenous? And when does that switch happen? It seems maybe there's not enough cohorts to look at that yet. I don't know.
No, I'm embarrassed because I should have reread these papers.
It's okay.
I can't remember off the top of my head.
Fine.
I want to say that they waited until the mother's antibodies had left I'm almost positive that they waited for a time point where the mother's passively transferred antibodies would not be present, and these were definite infant antibodies.
Do they have infants nurse if their mother is positive? I mean, is it automatic that it transfers transplacentally, or would they avoid them from potential? Is it in milk, and could it be transferred?
Yes, but almost all people are treated. I mean, you should be on antiretrovirals to prevent transmission from mother to child. But your question, there are numerous points at which the mother can transfer HIV to the child. I think it's getting your question, peripartum, postpartum.
Much
nursing.
Better said than I said. But yes, that's exactly
we are not in my field of expertise.
Sorry.
No, it's really
interesting. I know that Sally Permar's lab, but recently they published that they had matched the serum of mothers in the milk and then the serum of babies, and found that the escape mutants that were able to transfer to the babies were ones that were resistant to the mother's antibodies. So maybe they wouldn't even pass neutralizing antibodies, because the whole reason they're HIV infected is because they were resistant. I don't know.
Right.
Interesting.
No, it is interesting. Right. And I have to say, I don't focus on mother to child transmission, but you're right. And it has been shown that there's some pressure from the mothers, and, they are shaping the, virus environment or the immune environment, but they're shaping what virus would be passed on.
Right. There'd be like some selection pressure at that point.
Yes.
Now, this may not be a focus of your work, but maybe you could comment. I guess the goal is to try and make an antigen that will induce these, right?
Yes.
Is that possible, or do we have any leads?
Half glass full or empty?
Okay.
I believe it's possible. I think there have been great leaps and strides made in the past couple of years. I think all the strides made, everything is incremental and building up. So are we going to have it in the next couple of years? I might say no. Other people might disagree with me.
Yeah. Okay.
But I think just every year the B-cell technology and the structural technology, the ability to see the true structure of the proteins on HIV has really come such a long way, and that people have already translating that knowledge into different vaccine regimens. But, I think more has to be tried. And I think sort of whether you need to be targeting certain germline antibody, like you just are targeting certain antibody populations with your vaccine, or you're throwing in lots of different diverse antigens that represent lots of diverse viruses. I think people are pursuing all these strategies, and to me it's not clear which strategy is the correct strategy, but people are slowly improving the antibody response from where I would say vaccine trials were, 10 years ago.
Yeah. So you're probably familiar with this approach of giving a adeno-associated vector encoding a broadly neutralizing antibody, right?
Yes.
So what do you think about that? Is that viable?
That's a good question. I do think it's viable. I think most of the hurdles are, let's say clinical. I'm not sure if that's the right word, but how do you look at the anti-antibody responses? How do you play around with, fine tuning how much antibody is being given and where it's going, et cetera. But I think that's a really interesting strategy, because I do think that when we're doing the clinical trials right now that are sort of intravenous or sub-Q, you're always having the ability of ad-vectored antibodies to maintain a steady level of antibody versus the gradual waning, I think is a really interesting point that would be a pro for. Yeah.
But considering what you've already said, you probably would need more than one antibody, right?
Yes. So that's the other thing is I think we're at the very beginning of doing these trials where we're just testing methods, of ad vector delivery of antibody, but everyone takes it as a given. You need more than one antibody. And so how to deliver that, and then do you do in bispecific? There are all of these different formats in which you could give multiple antibodies. And so I think...
You want to talk about some of your work, what your interests are? I mean, we have been, but right now, what are you trying to do?
So right now, I'm still interested in antibody combinations for broadly neutralizing antibodies, as I had mentioned before. I've diversified my interests somewhat. Moving to George Washington University, there was a really strong cure research focus that I had never focused on before. So it's using trying different therapeutic strategies to see if you can cure people of HIV. So right now, HIV is very well managed and very well treated with antiretroviral medicine. However, if you stop taking these meds, your virus comes back. There's one person that has been cured of HIV, and that is only through transplantation. They had leukemia, I can't remember. So there's a really big effort in the field, and I was always focused on vaccines, and then sometimes I think what's harder, a vaccine against HIV or a cure for HIV, I'm not sure. But so that's another field that I've gotten more into with collaborators is how do you stimulate T cells that are latently infected with HIV to express antigens if you're going to use antibodies in cure strategies.
Yeah, we had a great talk here in I don't know if any of you went to the satellites. There's a satellite called Single Cell Virology on Saturday.
I didn't.
There's a talk from the Nussenzweig lab where they're trying to look at the latently infected pool and induce proviruses with drugs, right? And then see what do they look like. And they're clonal, right? And all of that. So it's really cool.
I think it's really cool, and I think that's really interesting. And I think that's another current hot topic in using antibodies against HIV.
Now, you've found escape mutations for VRC1, right? Have you tried the other broadly neutralizing antibodies?
Yes. So I've only so far analyzed in clinical trials data from people who are infused with VRC1 because there's still a limited number of antibodies that have been used in humans. But Nussenzweig's group has also done a couple of different clinical trials where they've reported the escape mutations from different antibodies. And we're trying to get an in vitro system where we can induce escape mutations in vitro to sort of broaden what we're looking at. But what we can say so far is that when you generate escape mutations, when you're giving these single antibodies, another question is, does that make these viruses resistant to more broadly, antibodies that target the same site? And with CD4 binding site antibodies, I think the answer is less clear. I think it's a complex epitope. And when we look at sensitivity to other antibodies that bind the receptor binding site, in general, the viruses are more resistant, but not always. And so I still am interested in if you took antibodies that see the same epitope, but in slightly different ways, can you box the virus in and limit how many changes you can make in the receptor binding site that the virus wouldn't be viable anymore.
In these clinical trials, are they administered to uninfected people? Is that how it works, or already infected? For
the safety profile, they administer to uninfected people, but the trials that I'm interested in where you can look at escape mutations, so a couple of them are people who are not on antiretrovirals, so they have actively replicating HIV.
Okay.
But also there have been a couple of trials where they've taken people who are fully suppressed, so they don't have replicating virus, and then give them the antibody and then take them off antiretrovirals to see if the antibody alone can suppress the virus. Mm-hmm. And again, so far, the rebound is slightly delayed, but so far it's, one antibody is not sufficient, which is, not surprising. And Nussenzweig's working on combinations, so I think that would be really interesting. But it appears, even though the virus isn't replicating, there's selection pressure. So the viruses that rebound appear to not be quite the same as just if you rebounded in the absence of antibodies. So that, so hopefully, if we can learn to apply enough selection pressure.
So one more follow-up, then you guys. Have they ever tried a protection experiment? So, just like the vaccine experiments where you give it to an at-risk population and send them out there, right?
Yeah, so there's currently I'm really interested in the results. It's called the AMP trial, which I believe stands for Antibody-Mediated Protection, where it's a very large trial in 2 sites, or more than 2 sites, but 2 areas. It's in North America and South Africa, where they've given VRC01 to people who are at high risk in those 2 different populations. Mm-hmm. And the results of the trial should be coming out the next couple of years. So I think that'll be really interesting.
How long does a bolus of antibody last? Do we know?
Yes. So so the regular antibody VRC01, I want to say the half-life is about 12 weeks. But there are mutations that you can make that now they're testing the profile of antibodies where you can mutate the FcRn binding site, and so the antibodies recycled so that you can play with the half-life and they call it the LS mutation. Right? So so you can play around and make it last longer. And
Thinking of broadly neutralizing antibodies from a vaccine standpoint, thinking about the immunology of why why there's a small percentage of people that produce these and some people none. Mm-hmm. So is it because you probably don't want a huge amount of broadly neutralizing antibodies because maybe that breaks tolerance? And so to actually overcome that, would you combine that with like a checkpoint inhibitor? I mean, at some point our body is saying, don't make these.
That's interesting. So I hadn't even sort of touched on that aspect. That's a really good point that people also talk about. Is there something about these antibodies that's necessary? The amount of somatic hypermutation, right. And even the epitope that they're seeing, that these are antibodies that are really autoimmune antibodies.
Like binding to your own sugars plus the HIV protein. Like, if would it start maybe an autoimmune situation?
Right. I don't think so. I don't think there's been any association with increased autoimmunity in HIV-infected people or people that make broadly neutralizing antibodies. There is an association with, I left off one epitope when we were discussing broadly neutralizing antibody epitopes, and that is what's called the EMPER region, which is the membrane proximal external region. So if you're looking at that glass picture of HIV, it's the stalk that's really near the virus membrane. And antibodies that see that, there's something about seeing that epitope in the context of a lipid membrane that is associated with autoreactivity. And so a lot of these antibodies, not all of them, but a lot of them that recognize that portion are polyreactive antibodies, which is, a question for vaccine research.
Yeah, if you're
Is it possible? Yeah.
Right, maybe we have a negative selection against those, but if you combined it with lifting some checkpoint, that's limiting that.
Right.
I don't know.
I tend to be more old-fashioned in that I just would induce antibodies against the receptor binding site.
Right,
but you're right. I mean, but that's a legitimate question. I think there's still so many questions to be answered about how to get an HIV vaccine, and that is, an area of pursuit.
Sure.
Is do you need to break tolerance, and is that the only way to get broadly neutralizing antibodies?
So you said that they have these trials going on both in South Africa and in the U.S., right? Mm-hmm. So are there different challenges in today's world about HIV, treating it, diagnosing it? Or what is different about the different places in the world and the challenges that are faced?
That's a great question. Kind of the caveat that I'm not one of those people who really, does the hard work of administering clinical trials. Right. But so there's geographic associations with different genetic subtypes. Mm-hmm. So, for example, one big difference in testing in North America and South Africa is the genetic makeup of the HIV that is currently circulating in that population. Mm-hmm. And so what's called subtype B HIV circulates in North America and Europe, and what's called subtype C is circulating in South Africa. And, when we say broadly neutralizing antibodies, not all broadly neutralizing antibodies are the same. Sometimes some antibodies are better against subtype C, sometimes antibodies are better against subtype B. VRC01 was isolated from someone from North America who was infected with the subtype B virus. So it's, important to think about these characteristics when you're, moving into clinical trials. And so I think having sites in different geographic locations automatically means you're looking at different genetic diversity. So that's really good for, I think, the realities of trying to develop a strategy to prevent HIV. But also, HIV is transmitted in different ways in different populations. And so the North American site might be targeting MSM, or men who have sex with men, which might be the population where HIV is more high risk from circulating for transmission in North America. And in South Africa, you might have different populations, might be more heterosexual transmission. And so I'd say the two main differences would be genetic HIV and transmission mode. So something else you need to keep in mind with clinical trials is the ethics now of testing vaccines, and people need access to antiretroviral. We have drugs that treat HIV. Right. And so counseling people, and the rules are different in different places as to how people go about getting access to antiretrovirals. So that would be something else that would affect how the clinical trial runs.
With working with macaques or monkeys, and the limitation now that there's a lot more regulation, I'm not sure, maybe Cindy, you know more about, they're moving towards not using monkeys in research, but what is your opinion? Is it a struggle? And
chimpanzees for absolute fact, right?
Yes.
Haven't they just basically eliminated their use? Because there's these new policies and permits that are required in order to do those kinds of primate studies?
Right. It's my understanding it's getting harder and harder. Most HIV vaccine animal model use rhesus macaques as opposed to chimpanzees. Okay. But I do know that it's getting harder to get rhesus macaques. And I also know that with the new interest in flavivirus, Zika and dengue, that I think people are competing for rhesus macaques in which to do animal model testing.
So are you saying the limited access is because of the competition or regulation or both?
I think it's more the regulation.
I see.
But that was just this, I have heard people complain about this. So that was just a side note. I don't know if that's really a factor. I think it's more the regulation. I think not only access to monkeys, but just in general, if you can get, high throughput in vitro systems as a way to test or to weed out different vaccine candidates. I think, everyone is interested in developing these new methodologies. I think you guys even might have talked about that in your RSV. Like yeast display is a big new technology that people are interested in. To just sort of in vitro, can you express different antibodies and just see which vaccine candidates bind? Yes, I think anything that can give you more information without doing expensive animal trials is useful to research and useful to the field in general.
And did your university, you have colonies? Do you have animal facilities right there at the medical school?
At George Washington, no, but actually I did my graduate school work at Yerkes Primate Center in Atlanta. So it was part of Emory, but there they did have colonies. They had a major colony that wasn't actually at the university, but the building we worked in did house animals, and there was a big neuro focus as well for the animals. And that was interesting. I got asked, people were very interested if you were anti-animal research before you worked there.
Oh, right, for the screening to make sure.
Right. And it was actually very civil. We would get these emails every now and then, on Saturday there's going to be a protest. So if you can work your experiments around it and don't have to come in. So there's a negotiation about when people are going to protest animal research. And
interesting.
Yeah. But there were lots of signs up all over about how, so many people are living because we've done animal research.
Right. As a way to kind of communicate the benefits in an effort to...
Which I think is important. I take it for granted because I believe so much in the science, and that, people have to justify the reasons, why you're using these animals and why they might die. And so it is thoughtful how people... it should be. It should be thoughtful how you use animals, but I also think it's necessary because you can learn so much from the immunology and vaccines in humans save lives. Right. And I think that's, the bottom line.
Absolutely.
Yeah. Although I have to say I hate doing mouse research. I don't know.
Do you look at the literature in other broadly neutralizing antibodies, like flu, for example?
It's hard to not. Like whether I want to or not. And I also joke that everyone in HIV is moving to flu because it's easier. I don't know. I don't know if that's true.
You might get an email from a influenza.
I know, I have to watch what I say. But there are a lot of people who were in HIV who have spread into different viruses like RSV or influenza. And I think no matter what virus you're looking at, that interaction between people who are interested in antibody responses and people who are interested in the protein structure of the virus, that sort of collaboration can manifest itself in different viruses. And it is interesting to hear about similarities and differences. Yeah, to me, it seems like anything else is easier from HIV.
So now that, you're within your first 5 years, what are some things as a new PI? What is something that was expected that, you love, that and then what is something that was surprising to you that was kind of, you had to adjust to?
Those are very good questions. So I would say the things that I love that may be expected is the science. When someone in your lab presents in lab meeting and they did some experiment that you had talked about before and it turns out how you think, or it turns out how you don't think and you brainstorm what happened, that's so cool. It's such a neat feeling to me, and I'm assuming to lots of people. And the downside, in my opinion, one of the hardest parts was learning to be a manager, that you are not, it's one, I think, very hard to let go of, I'm not doing the experiments, these other people are doing the experiments, and then how do I interact with them so that they do experiments either in the way I think they should be done, or I really want them to be able to articulate to me, because I might be wrong, which is, that's good, but I want them to be able to articulate to me why they did it differently, not that just, today's Tuesday and whatever, but that they're thinking, that they're thinking and thoughtful about it. And just basically people's personalities are so different. And so I've talked to lots of people, and I think, a lot of people find it challenging. How do you figure out who to hire? When you're starting out, you have limited budget, you want to be really careful and having people in your lab who are going to work hard, and you just want to get the most out of the money that you have and do the best science you can in this situation. So how do you hire people when no one's heard of your lab, and how do you manage them?
Right.
Yeah.
Was that something that you experienced as well when you both started your
I think
labs?
For me, and you also look very young. I think when I first had my lab, people were like, so whose lab are you in? I said, my lab. Yeah, but which lab is that? My lab. You're not a grad student? No. So that level of, and the other part of that is one, you're so close to the people in age with whom you are advising that but there's a power differential that is automatically comes when you have your new faculty position. And I think for me, one of the big things was when you're a postdoc in a lab, it's so collaborative, so interactive, so collegial, and everybody's sharing, and you're in the trenches together. And as soon as you move up one step, everybody's scared of you when you walk in the room. And so we're not scared necessarily, but it's everybody is on edge. So is she going to ask me a question? What I need to be looking busy or whatever. But it's definitely different. It's a different feel, and it's hard to get used to because you're like, no, I'm right there with you. I'm nice. I don't bite. I love the research. Talk to me. But it's different, and it's a challenge.
That's very true. I know, and I didn't. I had this past semester. I am trying to take undergrads, to get people in my lab who might last for a couple of years, and it's part of academic teaching. You want to involve students. And I had one undergraduate student who applied, and I guess I did not look at his date of birth on the resume and realized it was a veteran who was coming back to school. He was basically almost my age, and you just don't expect that with the undergrads. And it's fine, but you have that moment where you're like, oh, geez, we're the same age.
It's interesting. Do you do teaching as well?
I do. I have to say it's been a pretty good lenient start to my career. So I've had 2 full years as an assistant professor, and I've been able to slowly add in more teaching courses and get used to it. That's another difference. I wouldn't say I've enjoyed it, so it wasn't a problem, but it's very different from what I had been previously doing. And that's actually been kind of fun because, for example, looking for resources like your lectures online or just pictures from Janeway, it sort of makes you think and go back to, right, what are the basic mechanisms of all these things that I study? I don't think about B-cell development all the time, but it's good to remind myself and teach students. And actually, so I have a question for you guys. I was going to say the other thing about transitioning, if you're focusing on the transition from postdoc to PI at the NIH, because there's no grant writing requirement, I look back and laugh actually at my writing the K-22. Like I had some examples and I had great people to talk to, but I had no idea what I was doing.
No idea.
And so part of that transition is also budgeting, learning how to make a budget. It was incredible. That was out of left field for me.
Right. The only thing I can say is mentors.
Yeah.
Talk to people who've been through it, talk to people in your department, talk to colleagues inside and outside of the department, inside and outside of whatever university you're in, and get a wide array of different opinions and choose from those because they're all going to be different. Yeah. But I had exactly the same experience. I went through that workshop to write the grant as I was finishing up my postdoc because there was no example. Like my PI did not write grants. Like I had no guide. Right. And so I do take a lot of time with my trainees to have them get involved in the grant writing process. Not to get it out of my hands. I still do all the writing, but have them draft sections, have them look up things, have them write paragraphs of summarizing whatever it is that I'm interested in putting in the grant, to give them that experience. I have them read the grants. I have them help in the design process when we're first organizing the project and, what experiments are we going to do? What is a hypothesis? How do you craft a really good hypothesis? How do you craft the aims so that they make sense? Because we need to teach that.
That's actually great to hear because I'm hiring my first postdoc is starting in a couple of days. But I need to do that because that was something that I, right at the NIH you don't really get that chance to be trained in crafting grants, which is a whole new. Yeah, it was like a very large learning curve. And then I would just add to that I've always learned to ask people for templates of things, even just, I find it daunting sometimes where you have to, write a budget justification and you sort of know what you need to write, but what's it supposed to look like just to get different copies from people. And then you just figure it out and you have your own. Yeah.
I still do that.
Do you?
Oh, yeah.
Especially new things. So we're starting a project with collaborative cross mice. So I asked someone, send me your grant application. I don't know how to make the words right for this. I've never done it before. So why not? Yeah. You don't have to copy it, but
why
you can
would
learn.
You reinvent what's already available? You don't plagiarize, but you definitely use it.
And the words the concepts are so different that I'm not used to it. And it's hard enough, right? You might as well get a start. So I think that's good advice at any level.
Yeah. No, and I still have to tell myself because I do think there's something, I don't know, I personally have trouble asking for help, so I just have to remind myself.
Right now. Exactly. If you walk out of this room, don't be afraid to ask for help.
Right. No, I think that's really, really important advice. Yeah.
When I started, there was no internet or email. You had to call people, and it's harder. We had to, or go see someone, and it was a barrier for sure. But it's so easy now. You can email someone, and most people respond. It's pretty good.
It's true.
So that's a good advice overall, just to ask for help at any level, right?
All the time. Even if you don't think you need it, because you might realize there's something you hadn't thought of. I know I need it all the time.
With all aspects of everything.
Right.
I wanted to ask you that you're in this research center for the eradication of HIV, right?
Yes.
That's an interesting name, the eradication. So I work on a virus that's about to be eradicated.
Yeah. Right.
Polio.
That's right.
So I'm wondering if you think we can eradicate HIV.
I think it's a good goal. Theoretically, yes.
There's no animal reservoir. Right?
Well, there are,
of HIV, there SIV, but plenty.
Right. Okay, so
unless we have a new spillover, we can have a new spillover.
Right. So I feel like we have to acknowledge that.
And hopefully, we'll catch it earlier this time. Right.
Right, and we have drugs already.
Right. But yes, theoretically, it should be right.
Right.
I mean, if you could control everybody and stop transmission, then yes, right?
The problem is this latent reservoir.
Eventually, those latent reservoirs will die, right?
If you could prevent all new infections, right, then eventually
the population that has it will die.
But it would be like a generation or two.
It will, yeah.
Right. Yeah. And people have modeled, I think you can model that, if you reduce everyone, like if you treat everyone and reduce everyone so that they don't have any replicating virus, in theory, you just stop transmission, and exactly as you said. Yeah. I don't know in real life if, well, as you know from polio,
polio is easy. There's no latent reservoir. It's just an acute infection. It's
but just things happen in life.
Oh, yeah.
People, anti-vaccine campaign, you just can't predict.
Well, with polio, we have a vaccine that causes polio. So what do? You can't keep using it.
Right.
And as soon as you stop, you get polio. So it's a real conundrum.
Right.
We have to switch to an inactivated vaccine. But I think for HIV, probably antiretrovirals are not going to be enough to eradicate. Even though for Hep C, it seems that it will be, but there's no latent reservoir. It's an acute, and, a few hits with antivirals, and boom, it's way down. It's amazing.
Yeah.
Viral load is way down, but with HIV, you have this problem. So I think you need a vaccine of some kind.
That's probably true, and it's a debate in the field, especially, I guess, when you have limited resources where you devote resources, because really you should be looking at everything. Yeah. You should,
So do you sense that the intensity of the HIV research effort is still the same, or has it waned a little bit?
That's interesting. In my opinion, it's still the same, but everyone sees it wane. I've been warned so many times that it's not going to stay the same.
Yeah.
Like in the next 5 to 10 years, it is.
I have to say that if Tony Fauci leaves, that he's a big advocate for HIV research, right?
That is absolutely true.
I don't know who could replace him.
I think that is part of why people think
they're worried. Yeah.
Yeah.
Oh, right.
He's been an amazing advocate for HIV research.
I mean, as he should be, right? You need to have advocates, and you could argue that in this country it's somewhat under control, right? But
right.
Still, you need to help other countries, right?
Yeah, I think there are lots of arguments. We should help other countries. It's not entirely gone. Sure. And it also, you learn so much. It's still the technology and the thought process in investigating HIV vaccines is applicable to many, different viruses, and people then, you learn in HIV and have spread out. And so I think there's been a big boost in vaccine research overall against viruses right now that we'll see, the effects, the fruit.
Totally. That's research. A good point. Technology always bleeds over to other areas, right? So I was just at the Cold Spring Harbor Retrovirus Conference a few weeks ago, right? Did a podcast, and I said, who works on HIV? And 400 people in the room, they all raised their hands. Everybody. Who doesn't? A few people here and there. And so we had some older people on the panel. They remembered when there was no HIV. It was other retroviruses. But because of that, they were ready for HIV. They knew protease. They knew how to inhibit things, RT, integrase, right? All these things had already been discovered.
Right.
So that's an argument for doing basic science, right?
And how important it is.
And you don't have to have a translational goal in mind all the time. You could work on worms or mouse retroviruses, bird retroviruses. Nobody works on them anymore. It's a shame, right? Anyway, and you guys, anything else?
No, unless there's something else that's burning that you want to get out there about your program or your work, advice for grad students, any of that?
Yeah, maybe I didn't say this, but my other piece of advice that I was thinking about before this podcast is to have different mentors, to not only look for one mentor to fulfill your mentorship, but to have people in different stages of their careers mentor you and ask for help. Those are the two big pieces of advice.
You good?
I'm good.
Immune number 10. Where can you find it? You can find it at microbe.tv/immune, Apple Podcasts. If you have a mobile device, there's an app that will play podcasts. Just search for Immune. I'm pretty sure we're the only one that shows up. But if not, you can find us, Immune. Please subscribe. You get every episode free. And it helps us, if you subscribe, it helps us to have higher numbers, which gives us some flexibility. And if you have questions or comments, immune@microbe.tv. And of course, if you really like what we do, contribute. Go to microbe.tv/contribute. We have a number of ways To do that, you can buy a t-shirt, and right next to me, Cindy is wearing her immune t-shirt.
That's right.
Thank you. Thank you for your support.
I bought a mug too.
Cool. That's great. If someone told me I saw Cindy wearing her immune shirt, I said, great. Or you can do Patreon. You can give us $1 a month. That would be really cool. Microbe.tv/contribute. So our guest on number 10 of Immune today, our first in-person episode, has been Rebecca Lynch from George Washington University School of Medicine. Are you on Twitter?
No.
I tried looking for you to tag you.
No, you're way more advanced than I am on social media.
No. Twitter is gaining foothold here.
You should do it because it's a great place to see papers that other people are interested in.
Yeah.
Things that come up. It's great.
Yeah.
Anyway, thanks so much.
Yeah, thank you.
so much. I've really enjoyed
We
talking
appreciate
to you.
it. Cindy Leifer's at Cornell University. On Twitter, she's Cindy Leifer. Thanks, Cindy.
Thank you.
Steph Langel, Ohio State University. And I've met some of your colleagues here.
I know. They were excited to meet you.
And you as well. There's some Cornell people here.
a lot of Cornell
John
people
Parker here and
Yep.
a bunch of other people.
Colin
People
Parrish.
I've met on visits. Thank you, Steph.
Yeah. Thank
You are
all.
Stephanie Langel on Twitter. I'm Vincent Racaniello. You can find me at virology.ws, PROFVRR on Twitter. Music on Immune is by Steve Neal, stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.